Southern blotting, one of the most widely used techniques in molecular biology. Southern blotting is a simple mix of a few inventions by a scientist called E.M. Southern. Let us look at the learning outcomes of this session. So, Southern blotting as a technique is based on nucleic acid hybridization, wherein the target to be identified is a very specific DNA fragment or gene, or it can be many DNA fragments that have the same sequence or that have similar sequences. Southern blotting not only identifies DNA, but it also allows estimating the size of the DNA. The technique involves transfer of separated DNA from a gel to a nylon or nitrocellulose membrane, followed by hybridization with a probe. This step of transferring the DNA from a gel to nylon is why the process is called as blotting. The Southern blot analysis has found a lot of applications such as screening libraries for a clone, DNA fingerprinting, identification of methylated sites, etc. Now let us look at the discovery itself. So E.M. Southern, Edwin Southern, a molecular biologist, developed this blotting technique in the year 1975. He was actually working with rRNA genes and having isolated the DNA, he was looking for when he isolated the entire total DNA, he was trying to look at a way or trying to find out a way to isolate particular genes. He did try go in for density gradient centrifugation, but found it inadequate. So what he did was he isolated the DNA and then digested it with restriction endonuclease to get fragments. And those fragments then were subject to separation by agarose gel electrophoresis. Now, at this point of time, he faced another hurdle, and the hurdle was that he was left with the understanding that every separated fragment will have to be extracted from the gel one by one. And so he felt there has to be a better way of extracting these fragments from the agarose gel electrophoresis to a filter. In his own words, Southern stated, the prospect of cutting lots of gels into hundreds of slices, eluting the DNA from each of them and binding this to a filter that would then be hybridized and counted drove me to think that there had to be a better way of doing it. While searching for this better way, Southern invented his new blotting method. So what he did eventually is that this agarose gel electrophoresis was then blotted into a nylon membrane. So let us look at the general workflow of Southern blotting. DNA is to be isolated. That DNA has to be cut into fragments, then separated by gel electrophoresis, and those separated fragments have to be transferred to a membrane, which could either be a nitrocellulose membrane or a nylon membrane. And thereafter, the membrane has to be subjected to hybridization with a probe. And once the hybridization is done, then the detection of that hybridized probe would give a way of understanding which is the DNA to which the probe has bound. So DNA isolation, whether it is from bacteria or from plant cell or from animal cell, the treatment by which cell lysis and membrane disruption is done could be different. Once the cells have been lysed and the extract is obtained, the cell extract is obtained, it is subjected to deproteination. The general way of deproteination is using phenol chloroform. So with phenol chloroform, three phases are obtained. So there is a phase separation, and with the phase separation, the DNA and the RNA would be present in the aqueous layer, while the protein will exist as a buffy layer, and the organic layer of phenol chloroform will will be below, since the density is greater. The aqueous layer is collected, treated with RNase to remove all RNA. Thereafter, the aqueous layer is then subjected to precipitation by using either isopropyl alcohol or chilled ethanol. So that is how the total DNA is obtained. Now, this total DNA is then subjected to RE digestion, and with RE digestion, what one gets is fragments of the DNA. These fragments of DNA are then separated using gel electrophoresis. Generally, a molecular weight marker is loaded in one of the wells to kind of approximate the size of the fragments that have been separated. Once one has the separated fragments of DNA, they are then then they are transferred from the gel to a nitrocellulose membrane. This was a pre-publication sketch by Edwin Southern showing transfer of DNA from gel to the nylon membrane by capillary transfer. So I can see, as you can see over here, the gel is placed over here with paper wet with perspex and paper wet with the buffer. On top of it is present again a wet paper and then a lot of stacked dry papers. So the idea is that this gel, which has high content of water, and the dry filter papers which have less content of water will tend to move from the gel to the paper. So there will be a pull, or there will be a capillary transfer of the water from the gel to the paper, and since you have the nylon membrane present over here, or the nitrocellulose membrane present over here, the DNA from the gel will be pulled towards the nitrocellulose membrane because of the flow of the water towards the dry filter paper. Now, what was also done is that a pressure is applied over here to ensure that there is no diffused flow of the DNA from the gel to the filter paper. So therefore, this is how blotting is carried out, because the dry paper is going to soak the water from the gel. Now, before transferring, the DNA can be denatured by alkali treatment to get single-stranded DNA getting transferred. And then, once the nitrocellulose membrane has the DNA, it is baked at 80 degrees centigrade for 2 hours, or it is exposed to UV radiation. So, if it is nitrocellular membrane, then it is baked at 80 degrees, and if it is a nylon membrane, then it is exposed to UV radiation. Why does one do this? The main purpose of doing this is to enable the single-stranded DNA to cross-link with the nylon membrane, so its attachment to the membrane is firm. So this happens because the sugar phosphate backbone, which is negatively charged, interacts with the DNA, interacts with the positively charged components of the nylon or the nitrocellulose membrane. Now what is done is hybridization. And once you have the nitrocellulose membrane with the DNA fragments attached to it, from the gel it has got transferred to the nitrocellulose membrane. This membrane is now subjected to pre-hybridization treatment with a blocking agent. Say for example, bovine serum albumin is added. So all regions where there is no DNA gets blocked by the bovine serum albumin. The reason for doing this is that when hybridization is carried out with a probe, the probe does not have non-specific interaction with the other parts of the membrane. It would go and bind only to the region where it has complementary DNA. So the next step is hybridization with a single-stranded probe. So as you can see, you can have the probe getting attached to the membrane, but you can also have the probe binding very specifically to the DNA, again, DNA for which it has complementarity. So it goes like this. So once it is washed, what happens is that the non-specific interactions that were there of the probes with the membrane those probes move away, but the only probe remains bound, which has complementarity to the DNA. So say for example, this is the DNA and you have the probes coming and attaching. So you can see that this probe has less, affinity for the DNA, while these probes have more affinity for the DNA because of complementarity. You on washing, you can have this probe getting washed off, but these probes still remain attached because it has complementarity to these DNA. Now, the point is the attachment has happened, but how do you identify that there is a probe that has bound to the DNA? Which is why the probe has to be labeled. So, the probe can be labeled either with a radioactive component or with fluorescent tags. So, once they are labeled, then one has to detect where the label is present, because on identifying where the label is present, you automatically identify to which DNA it is bound. So after detection, one can find out that, say for example, this is the DNA to which the probe is bound, or you can have several DNA to which the probe binds. So this is an example of hybridized nitrocellulose membrane, and you can see over here how there are many DNA fragments to which the probe has bound, which means that all these different fragments have that sequence which is complementary to the sequence of the probe. So let us make the conclusions. DNA is isolated from a particular cell type or types, digested into fragments, and then separated using agarose gel electrophoresis. The separated DNA after alkali treatment are transferred to a nylon or nitrocellulose membrane and treated such that the single-stranded DNA is cross-linked to the membrane. The membrane is subject to pre-hybridization treatment to ensure that there is no non-specific binding, followed by hybridization with the specific probe. The sudden blot post-detection can be analyzed for identifying a single gene or many fragments having similar sequences. Edwin Southern integrated three inventions, restriction endonucleases, gel electrophoresis, and blotting, to create Southern blotting technique, which today has found several applications, including DNA fingerprinting. Thank you.